All right, it's my pleasure to introduce Ignacio Vasquez. He is a PhD candidate in biology at the Department of Ocean Sciences, Memorial University in Newfoundland, Canada. Ignacio completed his master's degree in aquaculture at Memorial University in 2022. His thesis focuses on Aeromonas salmonicida interaction with the environment and the fish host using genomics and transcriptomics analysis. Today, Ignacio will present one of his thesis chapters focused on dual RNA sequencing and host pathogen interaction. Thanks for being here.
Thank you for the oh, thank you for the introduction. So today I will talk about dual-seq transcriptomics of Aromonas salmonicida infection in Atlantic salmon primary macrophages that reveals lysosome and apoptosis impairments. And as an introduction, Aromonas salmonicida is one of the oldest known fish pathogens, and it's a gram-negative bacteria, non-motile with a wide host range, and is the causative agent of furunculosis. It's a disease that will end up in a systemic shock and organ collapse, and it is mainly affecting salmonids, but also currently it is affecting emerging aquaculture species like Atlantic cod, sablefish, and lungfish. However, very little is known about Aromonas salmonicida lifestyle. Thus, my thesis focus on fulfill all these knowledge gaps. And for instance, Aromonas salmonicida is able to survive up to 6 months in seawater. However, do not enter into a non-cultivable state and stay as a dormancy state. But however, the mechanisms that underlie to this ability to become in a dormancy state are currently unknown. And similarly, when we refer to the response of Aromonas to an external condition of the host or in the presence of the host are, are, are not yet explored. And in terms of the internal condition of the host, Aromonas salmonicida also the response is not well understood. And today I will focus mostly in the internal conditions and aspects of Aromonas salmonicida. So when we refer to an internal condition of the pathogen, we refer to several host pathogen interaction, and most of these interaction are going to be driven by the Aromonas has to deal with all the immune response from the host and gets into an intracellular level to hijack different pathways within the host cell. And most of these virulent factors and the mechanisms that Aromonas uses to immunosuppress the host are still unclear. So the aim of this study basically is to identify relevant genes that Aromonas salmonicida use in the pathogenesis in a real time in Atlantic salmon primary macrophages as the first lines of defense of the host. And for that, we use dual RNA sequencing technology to evaluate the host transcripts and the bacterial transcripts toward evaluated the host response to the infection and also the infection process at the same time. So first, what we did was basically to evaluate the infection process and using a gentamicin exclusion assay, where we took primary macrophages from the kidney of salmon and isolate them, wait until 5 days and until they are mature, then infect them with 10 to the 60 few per ml in a 1-to-1 ratio bacteria cell. And after 1 hour post-infection, we were able to recover about 6.7% of the attached bacteria cell. And once we add the gentamicin to eliminate external bacteria cell and just evaluate the intracellular one, we observe a percentage of recovery within 0.25 to 0.15% after 4 hours. And Additional to this, we observed that there is kind of a stable cell viability up to the end of the assay at 4 hours, as you can see here. I want to notice that if you look into this part of the panel, you can see a control non-infected cells where these black dots are going to represent melanin pigment, which is characteristic for Atlantic salmon macrophages. And after 2 hours, we start to observe an increased number of intracellular bacteria and also a considerable reduction of this melanin pigmentation that could be probably playing a role in the high phagocytic activity as we observe. Interestingly, when we look to the confocal microscopy for looking for Aromonas salmonicida intracellularly, we observed that at 2 hours post-infection, there is a cytoskeleton organization or actin filament reorganization that will merge with Aromonas, as you can see here. However, we don't know if this is going to be linked to phagocytosis or lysosome formation. And to get more deeper into this, we analyze the transcriptome profile of the host cell at 1 hour and 2 hour post-infection. And we observed that at 1 hour, there is about 871 differential species genes. And at 2 hours, there is an increased number of differential species genes, about 1,600, and where only 587 genes are going to be commonly dysregulated. And in the right side here, you can see mostly where all these genes are going to be associated with. And we found that they are associated to innate immune response, to intracellular signaling pathway, cellular differentiation, to apoptosis and cytoskeleton organization. And this also agreed with what we observed previously in our microscopy, electron microscopy, and the confocal microscope. So in terms of the bacterial transcriptional analysis, we use another approach to be able to enrich the bacterial transcript. So we took basically macrophages coming from a single individual and perform technical replicates to homogenize the response to the infection process. And we were able to enrich the bacterial transcript up to 0.055% of the total transcript obtained of the experiment. And as you can see, we were able to cluster the bacterial transcript per time point, as well as or similarly, we observe clusterization in the gene expression according to time points. And regarding to virulent factors, unluckily, we were not able to enrich virulence factor coming from the virulent plasmid. However, we found a few virulence factors that are going to be present coming from the chromosomal aspect, as well as it is the hemolysin AHS1, the heterolysin port toxin, and one of the genes that is type A, that is type 4 major pili. And these genes particularly are significantly expressed at 2 and 3 hours post-infection. However, even though we noticed some of these virulent factors, we still didn't know how the bacteria affects to the host cell in an intracellular state. So for that, we observe a differential expression of genes associated to early endosome and lysosome formation at 1 and 2 hours post-infection. And we decide to perform a lysosome acidification assay, as you can see here in the panel in the left side. So for this, we use a non-infected cell. We use a bacterium formalin-killed cell, which is the same Aromonas salmonicida, and our live bacteria. And as you can see, the formalin-killed bacteria induce or trigger a high lysosome formation compared to the live bacteria. And also, we start to observe a higher number of lysosomes becoming acidic and getting into the maturation process, in contrast to our live bacteria that it show a lower acidification in terms of lysosome. So these results also are going to suggest that Aeromonas could be modulating this lysosome maturation process to just stay intracellularly, but we still don't know if this is going to affect to the cell viability. And to overcome that issue, what we start to see was that there were some genes associated to an anti-apoptosis fashion that are particularly or partially inhibiting the apoptosis process in the host cell, as there is the expression of anti-apoptosis protein NRR13 or the apoptosis regulator Bakelin-2 after 2 hours post-infection. So we decided to perform a caspase-3 assay just to observe what was the activity in terms of apoptosis once that the cell would become infected. So as we did with the lysosome assay, we utilize non-infected cell as a negative control, the bacterium formalin kill as a positive control for apoptosis. This was previously observed in others experiments, and we use our live bacteria to just evaluate caspase-3 activity. And surprisingly, we noticed that and I suspect the bacterium formalin kill induce and trigger a more high caspase-3 activity, indicating a high number of apoptotic cells. But in contrast, the live bacteria, it seems to partially inhibit the apoptosis, even when we compare to the non-infected cells that show a kind of a basal caspase-3 activity, as you can see here in the flow cytometer results, and also in the confocal microscope. So as a take-home message of this dual RNA sequencing, it is a powerful tool that provide more context for the understanding of the host pathogen interaction. Also, the early infection of Aeromonas pseudomonas in primary macrophages aligned with previous characterized immune response in Atlantic salmon and other teleos. And also, Aeromonas can modulate the host defense during detachment by modulating the recognition and probably the cell death pathways. And this insights also suggest that Aeromonas become intracellularly by evading pattern recognition receptors using its A layer. And for the attachment, the di4-pili, and synthesizing and secreting hemolysin like HS1, and also the aerolysin for toxin ERE as the major virulence factors. And the transcription enrichment using macrophages coming from a single individual are going to improve the bacterial transcript homogeneity that enable also to identify key chromosomal virulence genes during this attachment and invasion process. And also the lysosomal and the caspase-3 activity analysis revealed to us that Aeromonas salmonicida modulates phagocytosis, apoptosis, and the immune response to facilitate these early stages of the infection. And with that, I would like to thank all the lab members and also the JBAR staff to keep and maintain the salmon and our CREATE network, and our external also collaborators coming from France and Brazil. And appreciate it.
Great. Thank you very much, Ignacio. Excellent presentation. It's a very interesting project. We're at time though, so folks, if you have questions, feel free to drop them into the chat. And Ignacio, if you can keep an eye there, maybe you'll have a chance to respond to them.
Yeah.
Thanks again.